Hello students, in this lecture I'm going to be covering parts of the cell, excluding the plasma membrane. And I'm going to start with talking about the nucleus and some of the roles that it plays in terms of cellular function. So the nucleus in a eukaryotic cell is the largest organelle, about 5 microns in diameter. Now some cells are either anuclear, such as the case with red blood cells, or multinucleate, such as skeletal muscle cells. Most cells, however, just have a single nucleus. So the nucleus also contains a nuclear envelope. Really, the nuclear envelope is a 2-unit membrane that is held together at nuclear pores. So it's essentially a double membrane that forms the outer boundary of the nucleus, and the nuclear pores will allow for material such as messenger RNA to exit the nucleus. The nucleoplasm contains the chromatin, which is thread-like material comprised of the DNA and proteins associated with the DNA, and the nucleoli include dark masses where the ribosomes are produced. So ribosomes I will be talking about more throughout this lecture, but they are the protein synthesizers of the cell. So if we look at some micrographs of the nucleus, these would be taken with an electron microscope. This first one shows the nuclear envelope as the boundary, the nucleoplasm as the kind of lighter colored inner material, and then the nucleolus as the darker material. And then the surface of the nucleus is shown in the second micrograph, and this is where the nuclear pores are visible. So again, the pores are basically just openings within that nuclear double unit membrane that will allow for passage of materials. So let's talk about a little bit of DNA's structure and function, because the nucleus is of course known as the command center of the cell. This is where the genome resides within a eukaryotic organism. So this is where all of the DNA that gives us our physical traits is going to be held. So DNA's purpose is ultimately to replicate itself. Basically, this is just making copies of itself. And this is what allows for mitosis, which is cell division, to occur. So replication is just the process of DNA producing 2 daughter strands from one original parent strand. And it relies on what's called complementary base pairing. Complementary base pairing is where the nitrogenous bases of the nucleotide, the A, T, C's, and G's, adenine, thymine, cytosine, and guanine, we have complementary base pairing, meaning the adenine is only going to bind with thymine, and the cytosine is only going to bind with guanine. So when you have a parent strand making this complete double helix, these little rungs of the ladder, I guess you could think of them as, are the nitrogenous bases. Those are the A, T, C's, and G's. So on one side of the parent strand, you might have an A, and the complementary side would have the T. Similarly, you might have C on one side, and then you would have to have G on the other side. So A always binds with T, and C always binds with G. Basically, when DNA replicates, you have a number of different enzymes that are acting to break the hydrogen bonds between the nitrogenous bases, and that opens up this double helix structure and allows for complementary base pairing to occur, such that the old strand can act as a template on which to build the new strand. So each new strand of DNA is going to be comprised of one original parent strand, shown in blue, and then a daughter strand, shown here in orange. And again, you're going to see that complementary base pairing. A always binds with T, C always binds with G. And this is what's called the semi-conservative model of replication. because you're conserving part of the parent strand when you're constructing each daughter strand. So of course we all know that genes reside within DNA. Genes are essentially just certain sequences of those A, T, C's, and G's that are the inherited directions for a cell or an organism. So there's several processes that occur in order for our cells to actually interpret the meaning of those A, T, C's, and G's, those specific nucleotide sequences within DNA. An organism's genotype can be defined as its genetic makeup. So that is just based on the sequence of nucleotide bases within the DNA molecule. And you can think of these bases as the functional unit of DNA. I kind of like to think of it as DNA has an alphabet of only 4 letters, and various combinations of those letters make up our genes. So that's the genotype, that's the physical molecular basis. And then the phenotype is going to be the organism's specific traits that are actually expressed. So how do we actually go from having this encoded information within our DNA to having physical traits? That's what I'm going to be talking about in the next couple of slides. All right, so this is going to be a very brief overview of these processes. If this were, a majors level biology course, we could spend a couple of weeks on the processes of DNA replication and transcription and translation, but I want to make sure that everybody knows the basics of these two processes, where they occur and why they occur. So essentially, the flow of information occurs from genotype to phenotype in 2 stages, transcription and translation. This is a simple illustration showing your DNA molecule within the nucleus of a eukaryotic cell. And remember that the nitrogenous bases, the A, T, C's, and G's, depending on what sequence they're in, they're ultimately going to determine what traits are expressed. So the information within the DNA is ultimately transcribed into what's called a messenger RNA molecule. So this is just labeled RNA, but really it should specifically say mRNA for messenger RNA. So this is another type of nucleic acid. It's an information storage molecule, and messenger RNA is using DNA as a template to take the message encoded in DNA outside of the nucleus. So mRNA has its own nucleotides, and the only difference in the nitrogenous bases is that mRNA has uracil in place of thymine. So essentially, the message that's held within DNA is going to be transcribed into that messenger RNA molecule. Messenger RNA is going to exit the nucleus via a nuclear pore, this opening within that double unit membrane. And once it's in the cytoplasm, it's going to come into contact with ribosomes and the cellular machinery that is able to actually translate that into a functional protein. So translation is when the message that's held within the RNA is made into a protein. So depending on the sequence of nitrogenous bases, different amino acids are going to be brought together in a certain order. So each of these little beads, these little beads on a string basically, represent a type of amino acid that has been bonded together with a peptide bond. And that is essentially how our genotype is transcribed and translated into our phenotype, because our phenotype, our physical traits, rely on protein structures within our bodies. So transcription can be defined as the transfer of genetic information from DNA into a messenger RNA or mRNA molecule, and a number of specific enzymes are involved here, such as RNA polymerase. So we have our double helix, our DNA strand here in blue. Enzymes are going to open that up, break the bonds between those complementary base pairs, and then the RNA nucleotides can be brought in as they are paired with ribose and phosphate groups, and they're going to do complementary base pairing. So very similar to what we see with DNA replication. Major difference is RNA has uracil in place of thymine. So where there's an adenine within the DNA molecule, a uracil is going to be brought in and it will pair with that. So transcription can occur. And again, basically this is taking the message that's within DNA, transcribing it into messenger RNA that can be taken outside of the nucleus. All right, so translation is the transfer of the information in messenger RNA into a protein. And this is when the language of DNA is translated into the language of polypeptides. So polypeptides are basically just protein strands. Poly means many. Peptides, think peptide bonds that form between the amino acids. And codons are defined as triplets of base pairs that code for specific amino acids. So let's look at this from top to bottom here. We have a DNA molecule, so double helix, deoxyribonucleic acid. The different colors represent different genetic sequences that we would refer to as genes. So if we just look at one portion of this, we have our DNA strand that has been opened up with the aid of enzymes. We have a sequence of nitrogenous bases here, AAA, CC, G, etc., etc. And then transcription is going to occur in which we have these complementary nucleotides of RNA using this DNA strand as a template. So messenger RNA is built off of that, and these triplets of base pairs, the base pairs are your complementary base pairing sets. Triplets would refer to, of course, 3. So essentially, what that means is 3 nucleotide base pairs are going to specify one type of amino acid. So this would be a codon, and depending on the sequence of bases in that codon, that's going to specify what amino acid is brought in with the aid of different forms of RNA within the cytoplasm. And those amino acids are going to be brought together with peptide bonds to actually form your polypeptide. So the primary sequence of a protein is just going to be your sequence of amino acids, and then depending on the conformation of the finished protein, that's where you actually get your functional protein, is the actual 3-dimensional shape of that protein. So ribosomes are the synthesizers of protein. They're found within the cytoplasm of cells, and they're also found attached to the rough endoplasmic reticulum. So they actually are the organelles that make synthesis of proteins possible. They contain ribosomal RNA or rRNA, and we also have transfer RNA involved here, or tRNA. Transfer RNA brings in the anticodons, which are bound to amino acids. So what this is showing is this whole 2-protein subunit here is your ribosome. The green is the transfer RNA, and the blue, the dark blue shapes represent the individual amino acids. So here's our messenger RNA strand again. There's some processing that's taken place here. Basically, these little end caps have been added, protective function here. This would represent a start codon, so that would essentially be a message that just tells the ribosome to start assembling here. And then from there on out, remember we have our triplets of base pairs that specify our amino acids. So you can actually kind of work backwards here and think about, okay, what does this actually mean here? Well, this would have been the first amino acid to be brought in during this translation process, and that would correspond with this first codon. And then this second shape course, this is just a cartoonish representation, but the second shape would represent a different type of amino acid that was brought in to represent or to pair with this codon. This third one would have been brought in to pair with that codon. And then this fourth amino acid right here is basically in the process of being added, corresponding to this codon. So depending on the specific sequence of nitrogenous bases, the A, U's, C's, and G's of messenger RNA, we're going to have different amino acids that are paired with those, and that actually makes your primary sequence in your polypeptide. So quick recap, I know this is a super quick treatment of these topics, but essentially we have this flow of information from DNA to RNA to proteins in the basically the functional blueprint of an organism is ultimately transcribed and translated here. So DNA itself, when we're just making copies of DNA, like in the case of mitosis, making genetically identical cells, DNA is copied using that process called replication. And then when we construct a messenger RNA molecule using DNA as a template, that is called transcription. And then when we're actually bringing in amino acids that pair with the codons within messenger RNA, that's a translation. So when DNA is transcribed, the result is a messenger RNA molecule, and then RNA is translated into a sequence of amino acids in a polypeptide. So it's also important to keep in mind where these processes occur. Transcription occurs within the nucleus of a cell. DNA is not going to leave the nucleus of the cell, so we're building our messenger RNA molecule there. And then translation is going to occur within the cytoplasm, so outside of the nucleus within the cellular contents where ribosomes are present. All right, so let's move on to other parts of the cell. I've just covered the nucleus and some of the important biological tasks that are associated with the nucleus and DNA. Let's move on to the cytoplasm. So cytoplasm basically just refers to all of the contents of the cell within the plasma membrane, and sometimes it excludes the nucleus. So organelles are little functional units of a eukaryotic cell that have specialized tasks to perform. Those that are bordered by a membrane are the membranous organelles, and these are only found in eukaryotic organisms. So the mitochondria, the lysosomes, peroxisomes, endoplasmic reticulum, and Golgi complex are all membranous organelles. And then organelles that are not bordered by a membrane include the ribosomes, there's many of them in an individual cell, centrosomes, centrioles, and basal bodies. We also have the cytoskeleton, which is essentially going to provide structure and basically a framework for the cell. The cytosol, which is the fluid within the cell. This is also known as the ICF, or intracellular fluid. And then we also will have inclusions within a cell. So inclusions are just stored products. They're not genetically coded for or constructed by the cell. They're things that a cell might engulf, dust particles, oil droplets, anything that's a stored product that the cell might take in. All right, so the cytoskeleton is a collection of filaments and tubules. These are proteinaceous, and they provide support, organization, and movement. They're comprised of microfilaments, actin specifically. This is the same motor protein that we see in our muscle cells that allow for contraction of muscles to occur. These microfilaments form a network on the cytoplasmic side of the plasma membrane called the membrane skeleton. and this supports phospholipids and microvilli and produces cell movement. And then we have intermediate fibers, which help to hold epithelial cells together. Epithelial cells are the cell types that make up our membranes and the linings of organs and our skin. So it helps hold epithelial cells together, resist stresses on cells, they line the nuclear envelope, and they also help toughen hair and nails. And then we also have microtubules. Might have gotten cut off there, but microtubules are also a component of the cytoskeleton. So the cytoskeleton, in a lot of depictions of cells within textbooks, you're not actually going to see these components clearly illustrated. They are the kind of framework for the cell itself. And you'll notice here that, you have some ribosomes basically just hanging out on these intermediate filaments here. So some of these protein tubes and fibers will actually help support organelles and provide for organelles to move around the cell, in addition to providing the basic structural integrity. All right, so let's look at the mitochondria. Mitochondrion would just be a single type of this organelle. Mitochondria would be many, so it's just the plural form. And if there's one organelle that students remember from their high school biology classes, it's usually the mitochondria. And it's because of the nickname that the mitochondria has gained, which is the powerhouse of the cell. That's because the mitochondria are involved in the process called cellular respiration, where we're converting energy stored in organic fuel molecules, basically the food we take in, converting that into cellular currency called ATP, or adenosine triphosphate. So mitochondria are membranous organelles. They have a double unit membrane similar to what we see with the nuclear envelope, and the inner membrane folds are called the cristae. The ATP is going to be synthesized by enzymes on the cristae from energy extracted from organic compounds. So our proteins, our carbohydrates, basically when we're breaking down any form of energy that we're taking in, we are going to convert that to ATP for use by our cells. And then the space between the cristae are called the spaces are called the matrix. This contains ribosomes and small circular DNA molecule called mitochondrial DNA. If you study cell types and the origins of life, you'll learn about the endosymbiosis, which has occurred between different cell types, has given us our complex eukaryotic cells. So basically, mitochondrial DNA is a vestige of this process. Mitochondria have their own set of DNA that's separate from the nuclear DNA of the cell. Moving on to the rough endoplasmic reticulum. The rough endoplasmic reticulum is parallel flattened membranous sacs that are covered with ribosomes. And it is continuous with the nuclear envelope and the smooth endoplasmic reticulum. So basically forms this big complex, which is interconnected by these membranes. What the rough ER does is it synthesizes packaged proteins. This is going to be important for our digestive glands. Phospholipids as well are synthesized, and proteins that are found within the plasma membrane are also synthesized here. The smooth endoplasmic reticulum is named because it does not have ribosomes like the rough ER does. The cisternae are more tubular and branching in the smooth ER, and this is where synthesis of membranes occurs, synthesis of steroids within the ovaries and the testes in animals, synthesis of lipids. Detoxification also occurs here, so this is important for liver and kidney function. And then calcium storage occurs here, which is really important for skeletal and cardiac muscle. So when we are studying the process of muscle contraction, we'll see that calcium plays a crucial role there. But it also has to be stored away, so it's only used during specific times. So smooth and rough ER, basically a depiction here. Rough endoplasmic reticulum is shown studded with ribosomes, but it is continuous with the smooth ER, so the membrane is continuous. And then some actual micrographs showing kind of like cross sections here. We have the nucleus of the cell illustrated, the cisternae forming these kind of maze-like labyrinthine networks here. Ribosomes of the rough ER, basically giving it this dotted look. And then smooth ER, similar looking, does not have those ribosomes, and this particular micrograph also illustrates an inclusion of an oil droplet. Okay, so ribosomes are granules of protein and RNA. They're found within the nucleoli, free within the cytosol, and within the rough endoplasmic reticulum. Ribosomes, as we saw earlier, use directions contained within the messenger RNA to assemble amino acids into proteins specified by the genetic code. Now, ribosomes, it's important to note that ribosomes are found in both eukaryotic and prokaryotic cells. So they are found in everything from bacteria to plants and animals, because all organisms are going to need to synthesize proteins. They're not a membranous organelle. They're a very simple, well, relatively simple, small organelle. The Golgi complex is a system of flattened sacs, cisternae. They synthesize carbohydrates, they package proteins and glycoproteins, which are complexes of carbohydrates and proteins. And they form vesicles. So vesicles include basically like little bubbles of membrane. They include lysosomes, secretory vesicles, so vesicles that will be secreted from the cell, and new plasma membrane. So Golgi complex, kind of a cartoon-like illustration showing some vesicles here, and then an actual micrograph. So a Golgi complex will have basically a receiving end and then a discharge end where it's receiving products, refining those products, and then sending those out, those finished products out to where they're needed within the cell or outside of the cell. So lysosomes are packages of enzymes in a single unit membrane. They are variable in shape, and the functions are for intracellular digestion of large molecules. A lot of people think of these as the stomachs of a cell. They perform autophagy. Auto means self. Phagy refers to consuming or eating, so autophagy, digestion of worn out organelles. Autolysis, which is programmed cell death. When cells are no longer needed, they will undergo this. And they will also break down stored glycogen within liver tissue in order to release glucose into the bloodstream. And then peroxisomes are going to resemble lysosomes, but they will contain different enzymes. Peroxisomes are produced by the endoplasmic reticulum. They're in all cells, but they're especially abundant within liver and kidney cells. So the functions of peroxisomes are to neutralize free radicals, detoxify alcohol and other drugs and toxins, as are found in the Kupffer cells in the liver. They use oxygen to oxidize organic molecules, which then will produce H2O2, hydrogen peroxide, as a byproduct. Catalase, which is an enzyme within the body, can then break down that hydrogen peroxide into non-toxic byproducts. And peroxisomes will also break down fatty acids into acetyl groups for use by the mitochondria. Okay, so finally, here's a look at some lysosomes and peroxisomes. So on the left here, we see lysosomes, which are somewhat variable in size and shape, but are generally kind of oval and small. This is actually a nice illustration of size and abundance of mitochondria as well. So, we're generally used to seeing these rather idealized illustrations of cells in textbooks that will usually just show like 1 or 2 of each organelle type. But in reality, an individual cell can contain many mitochondria. And they're not all these same kidney bean shaped structures. And then the peroxisomes, basically these bubble-like structures. So a lot of detoxification processes occur within the peroxisomes. All right, so that's going to wrap up the lecture over parts of the cell. Please let me know if you have any questions.